स्टूडेंट्स, वेलकम बैक टू योर बायो क्लास। सो हम सबको पता है कि प्लांट फिजियोलॉजी हम लोग ऑलरेडी कंप्लीट कर चुके हैं। तो सिलेबस का बहुत बड़ा पार्ट हमने कंप्लीट ऑलरेडी कर लिया है। तो अब हम स्टार्ट करेंगे ह्यूमन एनाटॉमी एंड फिजियोलॉजी, ठीक है, जिसका फर्स्ट चैप्टर है सर्कुलेटरी सिस्टम। अब सर्कुलेटरी सिस्टम का आप बेसिक तो पढ़ते आ रहे हैं जब आप छोटे थे तब से, कि वो deals with the heart, the blood and WBCs etc. Now in this chapter what you have already studied, right, we will discuss about the details. Okay, now to before starting with the circulatory system of humans, let us first get familiar with the body fluids. Okay, now body fluids are of three types. Those are the bloods, the lymph and the tissue fluids. Now blood तो हम सबको पता ही है कि that it flows through the veins and the arteries and is related with the actual circulatory system. Next comes the lymph. Now lymph is another fluid that flows along with the blood in another through another system, that is the lymphatic system. Now details about that we will discuss later in the chapter. Okay. Now the third is the tissue fluid. Now tissue fluid are the other fluids that are present in between the blood and the lymphs. Okay. Now there are two types of circulation, right? One is open type of circulation and is closed blood circulation. Okay. Now in humans we have the closed circulatory system, right? Otherwise blood तो इधर उधर कहीं भी बह जाता ना, तो बहता नहीं है क्योंकि it is closed. It is closed inside the veins and the artery system, right? तो but कुछ एनिमल्स में ये जो सर्कुलेटरी सिस्टम होता है वो ओपन होता है, जैसे कि फॉर एग्जांपल कॉकरोच. ओके, that is they don't have any channel or capillary through which the body fluids will flow through. Okay, good. Now there are also some fluids, those are non-circulating, like those are static, doesn't circulate anywhere. Suppose for example the visceral fluid and the vitreous chamber in the eye, the aqueous chamber in the eye, all of these doesn't circulate. Okay, good. Next what we will discuss in details is the blood. Okay, now blood we all know कि वो रेड कलर का होता है, वो आर्टरी और वेन से जाता है, ये सब तो हमको ऑलरेडी पता है. लेकिन हमने देखा है कि जो वेन्स होता है वो बाहर से ब्लू दिखता है, पर इसका मतलब क्या ये है कि ब्लड ब्लू होता है? Obviously not. The blood in the veins is actually dark red in color, right? And that is why sometimes after the layers of the muscles in the skin it looks or it seems to be blue. Okay, but actually में वो dark red color का होता है, और नॉर्मली तो ब्राइट रेड कलर का तो होता ही है. जो कुछ जान लेते हैं वो है द वॉल्यूम ऑफ द ब्लड. तो एन एवरेज ह्यूमन बीइंग कंटेन्स 5 to 6 लीटर्स ऑफ ब्लड इन इट्स बॉडी. ब्लड का टेस्ट तो वो सॉल्टिश टाइप का होता है, जो मेटल टाइप का टेस्ट होता है. ऑब्वियसली टेस्ट नहीं किया है पर होता है ऐसा, ठीक है. और शायद तुम कुछ लोगों ने ऐसा टेस्ट भी किया होगा क्या? एनीवेज, तो नेक्स्ट वी विल डिस्कस अबाउट द pH लेवल ऑफ द ब्लड. नाउ जो सॉल्टिश और जो मेटल टाइप का टेस्ट आता है ना, दैट इज बिकॉज ऑफ द अल्कलाइन नेचर ऑफ द ब्लड, एंड द pH ऑफ ब्लड इज 7.3 to 7.45. ओके, गॉट इट? वेरी गुड. नेक्स्ट कम्स द फंक्शन ऑफ द ब्लड. अब ब्लड का दो इम्पॉर्टेंट फंक्शन होता है, वो है ट्रांसपोर्टेशन एंड अनदर वन इज प्रोटेक्शन. ओके, तो ट्रांसपोर्टेशन तो हम सबको पता है कि जो डाइजेस्टेड फूड है, देन द ऑक्सीजन, द कार्बन डाइऑक्साइड एंड अदर मिनरल एलिमेंट्स, द एक्सक्रीटेड प्रोडक्ट्स, एवरीथिंग इज सर्कुलेटेड बाय दिस ब्लड ओनली थ्रू द सर्कुलेटरी सिस्टम. तो ट्रांसपोर्ट का तो हमको पता ही है. प्रोटेक्शन. Now protection, we know that blood contains white blood cells, that is WBC, which produces the antibodies, antitoxins, which helps in fighting diseases. Blood platelets help in clotting the blood. In this way, blood helps in protection in many ways. Okay, good. Now let us come to the composition of the blood. Now the composition of the blood is 55 to 60% plasma and 40 to 45% कॉर्पसेल्स, राइट। नाउ प्लाज्मा तो एक स्ट्रॉ कलर का लिक्विड होता है जिसमें इनऑर्गेनिक सॉल्ट्स, ग्लूकोज, एल्बुमिन्स, फिर हॉर्मोन्स, फिब्रिनोजन सब कुछ होता है, ठीक है। और कॉर्पसेल्स में क्या होता है, दोज आर द एक्चुअल सॉलिड एलिमेंट्स ऑफ द ब्लड which is composed of that RBC, WBC and platelets, right। now plasma विदाउट द फिब्रिनोजन, फिब्रिनोजन एक कंपोनेंट होता है, वो क्या होता है वो हम बाद में पढ़ेंगे। तो जो प्लाज्मा होता है, जब उसमें से फिब्रिनोजन निकाल लिया जाता है तो उसको हम सीरम कहते हैं। क्या कहते हैं? सीरम कहते हैं। सो प्लाज्मा माइनस फिब्रिनोजन इज इक्वल्स टू सीरम। नाउ कैसे इसे एक्सट्रैक्ट करते हैं वो तो हम लैब में ही कर सकते हैं, ठीक है ना। अच्छा, नाउ लेट अस डिस्कस अबाउट द ब्लड कॉर्पसेल्स। Those are of three types: the RBC, which is the red blood cell, also known as the erythrocytes. Next comes the WBCs or white blood cells, also known as leukocytes, and the platelets, also known as the thrombocytes, right? Let us first deal with the RBC, which is the red blood cell or erythrocytes, right? Now सबसे पहले जान लेते हैं कि आरबीसी का साइज कितना होता है। आरबीसी का साइज, हम तो आरबीसी का सेल्स तो ऐसे देख नहीं सकते, उसके लिए माइक्रोस्कोप की ज़रूरत होती है ना। तो उसका साइज इतना छोटा होता है कि हम उसे खुली आंखों से नहीं देख सकते। तो उसका साइज क्या होता है? ऑलमोस्ट अराउंड 7 माइक्रोन, ठीक है। नेक्स्ट, इन अ ब्लड, राइट, इन ब्लड देयर इज अबाउट 4.5 to 5 million RBC cells inside per cubic millimeter of blood। मतलब समझ रहे हो ना कि कितना होता है, बहुत सारा। तो नेक्स्ट कम्स द शेप ऑफ द आरबीसी। शेप ऑफ द आरबीसी तो सर्कुलर होता है, ठीक है। और अगर साइड से देखा जाए तो उसका शेप ऐसे बाइकॉन्केव दिखता है। क्यों दिखता है वो हम बाद में बोलते हैं, ठीक है। तो now the blood, which is the RBC actually, the RBC appears to be red, right? From the name only we can get it is the red blood cell। तो वो रेड कलर आता कहाँ से है? तो वो रेड कलर आता है हीमोग्लोबिन से। क्यों? जो हीमोग्लोबिन होता है, ऑल्सो रिप्रेजेंटेड बाई HB, उसमें होता है आयरन एंड प्रोटीन, ठीक है। तो इसी पिगमेंट के वजह से, विच इज द हीमोग्लोबिन, तो हीमोग्लोबिन की वजह से आरबीसी का कलर रेड होता है, ठीक है। नेक्स्ट, जब हम फंक्शंस में ऑलरेडी पढ़े थे कि दैट इट हेल्प्स, ब्लड हेल्प्स इन ट्रांसपोर्टिंग ऑक्सीजन एंड कार्बन डाइऑक्साइड, राइट। तो कैसे होता है देखते हैं। तो व्हेन hemoglobin attaches with the oxygen, okay, it carries the oxygen with itself, okay। So when the oxygen attaches itself with the hemoglobin, it becomes oxyhemoglobin or represented by Hb.O2, okay। Next, if the hemoglobin is attached with the carbon dioxide while carrying the carbon dioxide, it becomes carbaminohemoglobin, okay, which is represented by Hb.CO2, okay। Next comes the carboxyhemoglobin. Now what is carboxyhemoglobin? Carboxyhemoglobin is something when hemoglobin gets attached with carbon monoxide, okay, then it forms HbCO or carboxyhemoglobin. This situation is also known as carbon monoxide poisoning, since we all know that carbon monoxide is very injurious to health and is poisoning in nature, right। तो दोनों चीज में कंफ्यूज मत होना एग्जाम के टाइम लिखते वक्त कि कार्बन डाइऑक्साइड के साथ जब अटैच होता है तो उसे कहते हैं कार्बामिनोहेमोग्लोबिन, और कार्बन मोनोऑक्साइड के साथ जब अटैच होता है तो उसे कहते हैं कार्बोक्सीहीमोग्लोबिन, ओके। डोंट गेट कंफ्यूज बिटवीन दीज टू, ओके। नाउ लेट अस प्रोसीड। Now let us know that where these RBCs are produced, right? Now the RBCs, when we take birth, the RBCs are produced in the bone marrow, okay? This is a very important point. It can easily come in the exam. Now the mature RBC cells have no nucleus, no mitochondria and no endoplasmic reticulum or ER, okay? Now because the mature RBC cells doesn't have any nucleus, it appears to be hollow in the middle and biconcave from the side, right? Now, due to this absence of the nucleus, it can easily squeeze out through thin capillaries, veins, and arteries, right? Now, it also doesn't have mitochondria, which means that there will be no synthesis of ATP, right? And synthesis of ATP needs oxygen, okay? तो इसका मतलब क्या होता है? तो इसका मतलब होता है कि जब RBC cells ऑक्सीजन को लेता है, it doesn't uses the oxygen for its ATP production. पूरा जो ऑक्सीजन है वो बॉडी के सर्कुलेशन में काम में आता है, ठीक है? तो नो ऑक्सीजन is required by the RBC for the synthesis of ATP, right? Next, due to the absence of the endoplasmic reticulum, it can easily squeeze out through the capillaries, veins and arteries, right? ठीक है? Now, जान लेते हैं कि RBC cells कितने दिनों के लिए बचा रहता है. तो its lifespan is up to 120 days only. तो इसका मतलब ये है कि 120 डेज के बाद बॉडी का सारा ब्लड खत्म हो जाएगा. तो ऐसा तो नहीं होगा ना. अगर ऐसा होता तो हम कोई भी सर्वाइव नहीं कर पाते. तो whenever this 120 after 120 days a set of RBC cells are dead, another new set of RBC cells are produced at that moment only. Due to this continuous regulation of the RBC production, there is no shortage of RBC cells, although when it dies within 120 days, right? Good. Now the RBC cells are destroyed in the spleen, which is a lymphatic organ, which we will discuss later. Next, it can be also destroyed in the liver and the bone marrow, right? Now the iron part of the RBC cells are retained in the liver, right? While the rest of the part of the RBC is transferred into or converted into bilirubin and excreted as bile pigment. Okay, good. Now, there is two abnormalities of the RBC cells, which is the abnormal production of RBC cells. Now, the abnormal increase in RBC cells is known as पॉलीसीथेमिया. ठीक है, मतलब अगर एकदम अचानक से RBC का नंबर इतना बढ़ जाए तो उसे पॉलीसीथेमिया कहते हैं. ठीक है. नेक्स्ट, देन abnormally decrease in the RBC cells is known as the erythropenia. ठीक है, अगर घट जाए बहुत जल्दी RBC cells abnormally तो उसे erythropenia कहते हैं. ठीक है, अंडरस्टूड. आई थिंक इसमें कंफ्यूजन की कोई बात नहीं है. ठीक है, तो फिर लेट अस मूव ऑन विद द WBC cells. ओके, WBC पे आते हैं. ठीक है, तो WBC white blood cells or you can call it the leukocytes. What are those? First of all, we need to know that how much quantity it is present in the blood, right? So WBCs are around 4,000 to 8,000 millimeter cube per unit of blood. Okay, जो कि रेड ब्लड सेल्स के कंपैरिजन में बहुत ही कम होता है. ठीक है, तो the first thing is that the WBCs moves by diapedesis. Okay, so now what is diapedesis? Now diapedesis is the movement shown by amoeba, as you all know. That is, it extends its body and moves, right, by through expansion and contraction. So these WBCs moves through diapedesis. That is, they squeezes out or oozes out through the blood capillaries. Okay. Now, what are the main functions of WBCs? The main function of WBCs are the phagocytosis, the inflammation, and the antibodies. Right? Now, what is phagocytosis? Phagocytosis is the process through which WBCs engulf the bacteria or the germs. Right? You have all already read about how amoeba gets its nutrition. Right? It gets its nutrition by engulfing the food around it or surrounding it through phagocytosis. So, white blood cells also engulfs the bacteria and germs through phagocytosis. Right? Next comes the inflammation. Now, why is inflammation required, right? Now in the infected areas, the infection spreads very fast, right? The germ spreads very fast. So to limit that, ताकि वो ज़्यादा ना फैले, the WBC cells inflammate the area so that it does not spread into a larger area. So that is why WBC cells are really necessary for this purpose, right? तो उसी एरिया में पस फॉर्मेशन होता है जब WBC सेल्स जाके यहाँ पे इकट्ठे होते हैं इन्फ्लेमेटेड एरिया के पास. Now what is pus? Pus is nothing but the dead germs, bacteria and the dead WBC cells, that is, which are already used up in that area, right? Next comes the production of the antibodies. Now antibody deals with the immune system of the body. Like हमें जब भी कोई डिजीज़ होता है, जब हमारी तबीयत खराब होता है, तो antibodies are the main units that works for the immune system of the body. So the WBC cells are the ones who produce these antibodies, right? Okay, now the antibodies, we will discuss that about in the higher classes, right? Now what this antibody does, let me give you just a brief. Now antibodies produce antitoxins. Now antitoxins किस लिए? एंटीटॉक्सिन्स इसीलिए जब बॉडी में कोई टॉक्सिन इंट्रोड्यूस होता है थ्रू एनी वायरस और बैक्टीरिया और जर्म, तो उसको फाइट करने के लिए एंटीबॉडीज प्रोड्यूसेस एंटीटॉक्सिन्स इन ऑर्डर टू किल दैट टॉक्सिन. नेक्स्ट, let us discuss about the categories or the types of WBCs. Now there are two types of WBCs, that is, there are two categories. Those are the granulocytes or the granulated WBCs, which contains granules in their cytoplasm, and another one is the agranulocytes or the non-granular WBC cells, which does not contain any granules in their cytoplasm. So now granulocytes or the granular ones are divided into three types. Those are neutrophils, eosinophils and basophils. Now neutrophil has a three-lobed nucleus, right? And obviously the granules. Eosinophil have a bilobed nucleus, that is two lobes, and basophil contains a irregular number of lobes in its nucleus, right? Now neutrophils helps in engulfing the bacteria and germs, while eosinophils specifically fights against allergic reactions, that is, the allergic reactions eosinophils are found there, right, in order to fight that. Next, basophils. Basophils produce histamine, which helps in inflammation. Now inflammation की क्या ज़रूरत होता है, तो वो हम पढ़ चुके हैं already, right? Okay. Now agranulocytes or the non-granular ones are also divided into two types. Those are lymphocyte and the monocyte. Now lymphocyte is the main point or the main cell which helps in the production of antibody, or we can say directly that lymphocyte produces the antibody. Now lymphocytes are produced in bone marrow as well as the lymph vessels and the lymphatic organs like spleen, right? And about that lymphatic system we will discuss later in the chapter, okay, don't worry. Now there is monocyte, which is monocyte helps in the ingestion of the germs, okay. Monocytes also doesn't contain any granule. Now remember one thing, lymphocyte is the smallest WBC among all five of them, okay, don't forget. Now where are these produced? Like red blood cells, white blood cells are also produced in the red bone marrow. Its lifespan is about 2 weeks. Now you can see here the lifespan of WBC, which is 2 weeks, is really shorter in comparison to the RBC, which was 120 days, right? Now, there are two conditions, which are the abnormal increase of WBC and the abnormal decrease in WBC count, right? Now, the abnormal increase in WBC count leads to leukemia, right? You heard it right. Leukemia is actually what we know as blood cancer. बहुत लोगों को गलतफहमी ये है कि ल्यूकेमिया इज़ ड्यू टू द अफेक्टेड आरबीसीज और समथिंग, बट नो, वो आरबीसी की वजह से नहीं होता है. वाइट ब्लड सेल जब एबनॉर्मली नंबर में बढ़ जाता है तो उसे ल्यूकेमिया कहते हैं, विच इज़ द ब्लड कैंसर, राइट? नाउ ल्यूकेमिया इज द एबनॉर्मल और द ह्यूज डिक्रीज इन द count of WBC cells. Got it? Good. Now let us move on with the platelets. Now platelets doesn't have any definite shape. It almost looks like some crystals. Okay. Now number of platelets is that it's 2 to 4 lakhs per cubic millimeter of blood. Although it is more than the count of WBC cells, but it is less than the count of red blood cells. Next, platelets are produced from the megakaryocytes. Okay. Now, from megakaryocytes, it forms into fragments and platelets are formed. Okay. It is also formed in the spleen. Now, what is the lifespan of platelets? Now, lifespan of platelets is 3 to 5 days, which is extremely short. Obviously, because platelets' main function is blood clotting, and we doesn't always need clotting, right? So, its lifespan is about 3 to 4 days, which is the shortest among all the lifespan of the corpuscles. Okay. Now, it is also destroyed in the spleen only. Okay. Now, platelets, we know that it is found in the site of injuries where it forms the blood clotting, right? जब कट जाता है तो हमारा जो blood clot होता है, जो कभी कभी बोलते हैं कि देखो हमारा blood clot हो गया है, तो उसका वजह क्या होता है? वो है blood clotting, right? You can say blood coagulation. Now what happens, like what is the actual प्रिंसिपल ऑफ द फंक्शनिंग ऑफ द ब्लड क्लॉटिंग? मतलब कैसे होता है? हमें पता है कि हाँ, ब्लड प्लेटलेट की वजह से ब्लड क्लॉट हो जाता है जब हम कहीं पर कट जाता है या फिर छिल जाता है, ठीक है ना? लेकिन कैसे होता है? व्हाट इज गोइंग ऑन, राइट? व्हाट इज द प्रोसेस? सो जान लेते हैं, राइट? नाउ द प्लेटलेट कंटेन्स thrombokinase. Okay. Now that thrombokinase is at first converted into thromboplastin. ठीक है, मतलब जब ब्रेन सिग्नल देता है कि कहीं पे कट गया है तो तब thrombokinase is converted into thromboplastin. Next, the prothrombin is converted into thrombin by the help of thromboplastin, which was formed before. So thromboplastin then converts prothrombin, which is inactive in nature, into thrombin, which is active in nature. Next, this thrombin, which is active in nature and formed, it along with the calcium ions converts fibrinogen into fibrin, right? So the thrombin along with calcium ions converts fibrinogen into fibrin. Now these fibrin rushes to the spot where there is a cut area or an inflamed area and forms threads or mesh around the area in order to cover the wound, right? And that mesh or the thread-like web-like structure catches the RBC cells and the WBC cells in order to heal that area, and the regrowth of cells is also occurring under that mesh only, right? Now after it has healed, right, the mesh forms a clot, which is the solid substance. जब कटा हुआ एरिया सूख जाता है, जब जो काला टाइप का चीज़ फॉर्म होता है उसे क्लॉट कहते हैं, और वी कैन ऑल्सो कॉल इट थ्रोम्बस. एंड watery like fluid oozes out around that area sometimes. That fluid is nothing but the serum. Now what is serum? Serum is already we have learned, that is the plasma without the fibrinogen. Okay, good. Now if there is an excess fall in the count of platelets, then it can lead to hemorrhages, okay, which can also cause death. Okay, now this situation, that is a huge fall in the platelet count, is caused by dengue. We all know that, क्योंकि हम सब ने बहुत बार सुना है कि देखो ब्लड प्लेटलेट का काउंट बहुत कम गया है डेंगू की वजह से. तो and why is that fatal? That is because it can cause hemorrhage, and hemorrhage is a fatal thing and it can lead to death. Okay, got it? I hope every concept is clear. WBC, RBC and platelet, everything should be clear for you now. Okay, I will also attach some other examples and details in the notes too. Okay, next we will discuss about the blood groups. So now let us discuss about the blood transfusion and the blood groups. Now blood transfusion means that is the donation of the blood so that another person can take that blood in case of if there is a shortage of blood in some other person's body, right? That is the introduction of someone's blood in another person's blood is known as blood transfusion. Now before moving on with the blood transfusion, let us discuss about the blood groups. Now blood groups are formed on the basis of the occurrence of antigens and antibodies. Now antigens are what? Antigens are the types of proteins present on the RBC cells, and antibodies are actually the alternatives of this antigen which actually fights against diseases, right? Because it is included in the body's immune system. Now antigens are of two types, so that are A and B, and also antibodies are also two types, that are antibody A and antibody B. Now how is it arranged? Let us see. At first we all know that there are four types of blood group, right? That is A, blood group B, blood group AB and blood group O. Now blood group A has antigen A but antibody B. That is, antibodies are the alternatives, right? So blood group A have antigen A and antibody B, whereas blood group B has antigen B and antibody A. Now the interesting thing about blood group AB and O is that blood group AB has both the antigens, that is antigen A and antigen B, but it does not contain any antibody. Next comes the blood group O. Now blood group O contains no antigen, okay, but it contains both the antibodies. Those are antibody B and antibody A. Now blood group AB is known as the universal recipient. Why? Because blood group AB can receive blood from all of the other blood groups, that is blood group AB itself, from blood group A, from blood group B and also from blood group O. Since it can take blood from all the other groups, it is known as universal recipient. Similarly, blood group O is known as universal donor, right? Because it can donate to all the other blood groups, that is blood group O, blood group AB, blood group B and blood group A. But blood group AB can only give blood to AB, whereas blood group O can only take blood from blood group O itself, right? ऐसा हो गया ना कि AB बहुत सेल्फिश है और O इज़ टोटली सेल्फलेस. उसे कोई दुनियादारी पसंद नहीं है, वो सिर्फ डोनेट ही करता रहता है. तो आई थिंक ब्लड ग्रुप O इज़ अ गुड पर्सन, राइट? गुड. Now blood group A can receive and give blood to blood group AB and blood group A, and blood group B can receive and donate blood to AB and B itself. Okay, understood? I hope the concept is totally clear. Good. Next comes the Rh factor or the rhesus factor. Now हम सब ने सुना होगा कि लोग बोलते हैं कि मेरा ब्लड ग्रुप बी पॉजिटिव है, मेरा ब्लड ग्रुप ए नेगेटिव है और ओ नेगेटिव है. तो व्हाट इज दिस पॉजिटिव एंड नेगेटिव, राइट? नाउ दिस पॉजिटिव एंड नेगेटिव कम्स फ्रॉम दिस RH factor or the rhesus factor. Now why the name rhesus? Now rhesus is the name because this factor was first found from a rhesus monkey, so it was named as a rhesus factor. Now if this rhesus factor is present on the RBC, then it is said to be positive. But if the rhesus factor is absent from the RBC cells, then it is said to be negative. Okay, now let's talk about the blood transfusion. Okay, now what happens in blood transfusion is that there should always be a compatibility, right? That I talked about, that A can only donate to A and AB, whereas O can donate to everyone, right? So that is the compatibility. Now if some blood group can donate to the other blood group, then those blood groups are said to be compatible. But if they cannot donate to each other, then they are called non-compatible, right? Now according to this compatibility, the transfusion is done, right? Now what happens in rare cases, like if RH positive is donated to RH negative. No, first of all, let us know that RH positive can donate to RH positive only and RH negative can be donated to RH negative only. So what happens here is that if RH positive blood is introduced into some person who has a RH negative blood, what will happen is that antibodies against RH positive blood is produced in the body of the RH negative person, right? Now what happens is that during the second transfusion, that is when the second time the RH positive blood is introduced in this person's body, the previously produced antibodies will totally fight against the RH positive blood, because वो सोचेंगे कि वो उसका दुश्मन है, right? So what they will do, that is they will clot the blood, right? So what they will do is that they will clot the blood of that RH positive one. In that case, it can be leading to fatal conditions, even death. Okay, clear? Now this is something that happens during pregnancy too. If the mother has a RH negative blood, what happens is that if the mother has a RH negative blood and she conceives a RH positive child, the first child will create no problem, like both will be healthy, right? But as we have learned here that in the first time it will create an antibody. So when this RH negative mother conceives a second RH positive child, and what happens is that the blood of the mother has that antibody for RH positive. Now that antibody identifies the RH positive blood as its enemy, and at the end it leads to the clotting of that blood of the fetus, which means that the fetus will obviously might die, and it can also lead to complications in the mother, right? So it should be always tested that is the child positive or is the child RH negative, if the mother is RH negative. Otherwise it becomes a risk for both the child and the mother, and it may cause the death of the child also. Okay, clearly understood? I hope you don't have any problem with this. Now the second part, we will deal with the actual circulatory system of the blood, that is how the blood flows around the body through the heart and the blood vessels. Okay, thank you for coming.